We're thrilled to welcome Fabian Gerlinghaus, CEO and co-founder of Solaris, to the show today. Thank you once again, Fabian, for joining us.
Thank you so much for inviting me. It's a pleasure to be here.
Let's kick things off. Fabian, can you share a brief personal intro with us?
Happy to. My name is Fabian Gerlinghaus. I'm the co-founder and CEO of Solaris, a life science technology company that's accelerating access to life-saving cell therapies. I'm originally trained as an aerospace engineer in Germany and then came over to the Bay Area, California, about 10 years ago and made a switch into life science technologies at that time when I joined Synthego. I joined Synthego when we had about 5 people in the garage. Synthego is a leading CRISPR-Cas9 genome engineering company, and then helped the company grow from 5 people to 230 people over a 5.5-year tenure. So just before starting Solaris, I was the Chief Innovation Officer at Synthego. Then in 2009, I started Solaris as I had become very aware of the problems in cell and gene therapy.
Thank you, Fabian. And before we dive too deep, can you just provide a brief intro to Solaris for us?
Solaris is accelerating access to life-saving cell therapies. So what's blocking access to cell therapies today is that these personalized medicines are very expensive and very, very difficult to produce at large scale. That's because we're talking about personalized, individualized treatments where we're, for the most part, making one cell therapy dose for every patient individually. And these doses are produced with a lot of manual labor. And what we're doing at Solaris is we're building a fully automated and closed manufacturing platform that fully automates the entire cell therapy manufacturing process for cell therapies, while also providing incredibly high throughput. So the impact of that work is that we're enabling cell therapy manufacturing in a way that is robust and reliable, very cost efficient, which hopefully will lead to reduced pricing for cell therapy drugs on the market. And above all, we're enabling the industry to scale out and actually meet patient demand for these life-saving therapeutics in the tens of thousands and hundreds of thousands of patient doses being produced per year per drug.
Well, Fabian, thank you for sharing that introduction to yourself and the company. We're really, really excited to dive in. And so to get us going, can you share a bit more about the founding story of Solaris, and was there a specific spark that started everything?
Yeah, so in 2017 and 2018, I was doing a lot of market research, more focused on the traditional biologics space. And a lot of the conferences that covered the biologics space also covered the newly emerging cell therapy space. And if you listened at any of those conferences, people were shouting from the rooftops, we need closed and automated cell therapy manufacturing technologies in order for cell therapy to become a commercially relevant treatment modality that payers in the payment system can actually pay for, patients can afford, and to solve the manufacturing bottleneck, to actually make these cell therapies manufacturable at large scale. And at that point, I decided to start Solaris in 2019 to build a cell therapy manufacturing platform that actually solves these problems.
Well, that's a perfect segue to our next section. We would love to chat a bit more about the technology that you're developing. But first, can we take a step back? You had mentioned that there were a list of challenges about the cell manufacturing and specifically challenges relating to the logistical intensity of it. Can you provide a bit more background on these manufacturing challenges that Solaris is aiming to go after?
Absolutely. So the way cell therapies, and in particular, I'm going to focus on autologous cell therapies for right now, are being manufactured today is in a process that starts and stops with the patient. So as the first step, you're collecting an apheresis product, a leukapheresis, that's a blood product from the patient. You're then shipping that to today, typically a centralized manufacturing facility where you use the blood product from the patient as the starting material for a very labor-intensive, complicated process in which you've got a team of highly trained scientists spend on the order of 2 to 3 weeks in very expensive clean rooms and they execute the entire manufacturing process that involves multiple unit operations on benchtop equipment that was never purpose-built for cell therapy manufacturing. All of these benchtop instruments typically handle one unit operation for one patient at a time. And in between, you're stuck with a manufacturing paradigm that is so manual that you end up having on the order of 50 manual processing steps per patient dose, about 80 hours of touch time. And these are highly qualified personnel, right, to produce one cell therapy dose at the end of this process. So the logistics of scaling this out to, say, 10,000 patient doses produced per year per drug are mind-boggling. The logistics really quite literally involve copy and pasting this very personalized and very manual process 10,000 times. So that means you end up starting 30 of these processes every single day. You end up running 400 of those processes in parallel every single day. And you need to hire more than 1,000 employees in manufacturing alone, which is extremely difficult to do in our industry today, given that there's a dearth of cell therapy, of qualified cell therapy talent. It's also difficult to train all of those people, right? It takes about 6 months to train somebody on GMP manufacturing methods. And the average retention time of qualified GMP personnel is about 1.5 years. So you spend 6 months training them and a year later they're gone. That's not efficient at all. So as if that wasn't enough, while you're going through this very complex manufacturing process, companies today generate between 400 and 600 pages of documentation in batch records. These are typically paper-based batch records. Just imagine that. We're quite literally, as an industry, writing a book in paper-based batch records for every single dose of a cell therapy that's being produced for every patient individually. And
I absolutely don't want to imagine that.
It's a lot of work. And again, you do that 10,000 times, now you're writing 10,000 books a year for every dose individually. And then on top of that, in all of these manual processing steps, you've got the risk of operator error. You can do as much training as you want. Humans are not good at repeating the same processing steps over and over again and never making a mistake. On top of that, you've got the risk of contamination because the process is typically open. That is because the only way you actually get the flexibility today to manufacture the particular cell therapy that you have developed as a pharma company is with benchtop instrumentation that handles one unit operation for one patient at a time. Now you've got all the flexibility in the world. You can tune and tweak all the process parameters, but you're locked into a manufacturing paradigm that is open and manual. And specifically when it comes to the openness of the problem of the process, the problem is the risk of contamination. Every time you move the cells from one of those single-use consumables made to, say, work with a centrifuge, to the next consumable made to, say, work with your cell sorter, to the consumable of your electroporator, to the consumable of your bioreactor, and all of these open transfer steps where you're moving the cells from one consumable to the next consumable, you've got the risk of contaminating your product. So 10,000 individual processes with all of these manual steps, all of these open transfer steps, manual sampling steps. That's a lot of opportunities for operator error. That's a lot of opportunities for contamination. And that's why the process failure rates that we're seeing in the industry are so incredibly high. Recently, there was an article that quoted a pharma company that said, we've got 18% process failure rates. First of all, that's huge compared to any other biopharmaceutical manufacturing process. And then it's not just an economic concern. When you're making a cell therapy product, it's important to remember that most of the time we're making these products in the field of oncology for terminally ill cancer patients. So you're quite literally carrying a patient's life in your hands as you're going through the process of making a cell therapy for them. That is oftentimes their last hope. It's oftentimes third or fourth line of treatment when they get to cell therapy. At least that's the case today. And if you mess it up, you may just not get a second chance of producing a high quality cell therapy product for that patient before the patient dies. getting to the point where we've got reliable processes is very important for patients. And the other point to make here is, as an industry, we're not meeting patient demand today. There was just last week an article in Stat that talked about the inability of pharma companies to produce enough cell therapies to meet the demand of all of their patients, which then means they're allotting a certain contingent, a certain number of cell therapy manufacturing slots to every cancer center. And that then puts the oncologists, the clinicians, in the position where they have to choose which of my patients am I going to give these limited number of CAR T-cell manufacturing slots to. And as a matter of fact, I was also, I'm quoting this article again, 20% of the patients that are eligible for CAR T-cell therapies die before they get a manufacturing slot, before we can make the cell therapies for them. So that's the state of the industry today. And that's why it's of paramount importance to actually make cell therapy manufacturing technologies that are reliable and robust, cost efficient, and also scalable.
What a fantastic overview for anyone listening if they weren't abundantly clear of really how challenging and important this topic is now. Hopefully it's made more than clear. So the next natural question to ask is, what is Solaris doing? How are you approaching these abundances of challenges?
Our manufacturing platform really consists of a single-use consumable cartridge that fully automates and closes the process. It then further consists of the cell shuttle. The cell shuttle is effectively a factory in a box that has the ability to run 10 processes on 10 of these cartridges simultaneously, and a very powerful software solution that gives pharma companies the flexibility to design their specific cell therapy manufacturing workflows in software and customize the platform to their specific process. So customization of our platform really happens in software, and that's how we're enabling the flexibility to run both autologous and allogeneic cell therapies, as well as many different cell therapy modalities, CAR-Ts, TCRs, NKs, basically any cell type that is a suspension cell type. So diving in a little bit more detail into a little bit more detail with the consumable cartridge, that is really how we closed and automated the cell therapy manufacturing workflow from start to finish. So in order to mitigate the risk of contamination, you need to eliminate any kind of open transfer steps from one consumable made by one vendor to work with one instrument to the next consumable made by another vendor to work with another instrument. So that's what we've done in our single-use consumable cartridge. That cartridge actually contains all of the consumable modules that are required for all of the cell therapy manufacturing unit operations, including enrichment, activation, cell selection, gene transfer with viral and non-viral methods, cell expansion, fill finish, and formulation. So it's an all-in-one consumable cartridge, and all of these consumable modules for the different unit operations are fluidically connected in a closed tubing set. That closed tubing set is then further protected by a hard shell that is made for automation. So that's how we're closing the process, mitigating the risk for contamination, and automating the process from start to finish, thus mitigating the risk of operator error. So that's the heart of the system. And the cell shuttle is a very sizable instrument. It's effectively a factory in a box. It's taller than I am. It's about 9 feet tall. It's about 15 feet by 18 feet at the moment. So it's the size of a room, and it maintains its own clean room environment. So you can think of the cell shuttle like a factory in a box that actually maintains its clean room like a clean room environment, like any modular clean room that's out there on the market today. And then within this clean room, you've got a robotic work cell that is actually serving instruments on both sides. So there are a number of bioprocessing instruments for all of the different unit operations, all of which are capable of receiving our consumable cartridge. And importantly, you can load up to 10 of our single-use cartridges into the cell shuttle at the same time. So that's an order of magnitude improvement in instrument throughput compared to any other technology that's out there on the market today. So big picture, by closing and automating the process, we're eliminating opportunities for process failure due to contamination and operator error. And the net result of that is that we're bringing down the process failure rates by at least a factor of 3, possibly more. Then by eliminating a lot of the human labor, which is actually, it's great for the humans too, because who wants to do repetitive work in a clean room? Like especially today, all of these personnel, all of the staff that's doing cell therapy manufacturing today, are usually highly trained scientists that would be much better leveraged doing process development, developing new cell therapies. So it's also a necessity to automate the process if you want to make it scalable and actually save more patient lives. So by automating the process and by virtue of the fact that we're about 80% more space efficient with our technology compared to manual cell therapy manufacturing methods, you can now build much smaller facilities with way less people to achieve the same patient throughput per year per drug. That ultimately makes for much lower drug prices. So we're lowering cell therapy manufacturing costs on a per patient basis by up to 70% compared with manual manufacturing paradigms. And then the last point that's really important here is scalability. The industry today is not meeting patient demand due to the lack of automated and high throughput manufacturing technologies. And that's what we're building. So when I say 20% of the patients that are eligible for CAR T cell therapies are dying before they get a manufacturing slot, that is the problem that this technology can solve. And if you're asking me, well, what makes it so scalable? The quick answer to that is really most of the manufacturing equipment that's on the market today handles 1 unit operation for 1 patient at a time with tons of human labor in between. And what we're building is a platform that actually handles all of the unit operations, the entire cell therapy manufacturing process for up to 10 patients simultaneously. And by fully automating and providing really high throughput, we've created a very scalable manufacturing platform that actually enables commercial scale manufacturing in the tens and hundreds of thousands of doses per year per drug.
And talk about an incredible breakthrough, especially when you're thinking about, as you described, providing access to patients who otherwise wouldn't really be able to even test the therapies on the market, never mind those incoming. So I think from all of us in the patient-facing community, we're excited to see the scale and excited to see what's built.
Absolutely. And the problem, like the pain of not having an answer to commercial scale manufacturing, it's only going to get much more severe, given that there's more than 700 cell therapy companies developing new cell therapies in the world today. So we've really seen an explosion of activity in the cell therapy world. And with all of these drugs in the clinic, the FDA made a projection a few years ago that they'll be approving 10 to 20 new cell and gene therapies every single year starting 2025. So there's a lot of cell therapies coming down the pipe, and manufacturing is an unresolved problem. So that's what we're here to do.
And as you talked about and as you described, there's not only the challenges around costs, but there's also the challenges around development that you're just talking about now. So let's take this a step back. How will Solaris technology impact the earlier R&D timelines for cell therapy?
Absolutely. So the beauty of the whole cell therapy paradigm is that we're not scaling up, we're scaling out. So what I mean by that is when you're making monoclonal antibodies, you go to larger and larger bioreactors. You start at the 1 liter scale, then you go 50, 500, 2,000 liter bioreactors, and you go up. And every time you do that, every time you go from one scale to the next scale, you need to do process development. You need to do comparability studies and make sure you get the same results, the same titer. In cell therapy, that's not the case because autologous cell therapies, we're basically making 1 drug for 1 patient. So we're staying at effectively the same range of volumes throughout the entire drug development process, from preclinical development to clinical development to commercial scale development. So what that means, and this is certainly something that we took into account when we designed the cell shuttle, is we can build a platform that is very, very valuable throughout all drug development phases. The high throughput of the platform that I just talked about is obviously tremendously valuable in the context of commercial scale manufacturing, when your process is fully defined and you just want to run the same process over and over again to reach as many patients as possible. It's actually also really valuable as a high throughput preclinical process development tool. So late in the preclinical process development phase, you can do design of experiment studies where you define in software not just one recipe, but 10 different workflows with 10 different permutations of process parameters, possibly different reagent choices. And in this way, you run design of experiment studies to determine what's the best recipe for my particular cell therapy workflow. Importantly, you can do that. You don't need to do that on a full scale. You can work with, say, a quarter or a fifth of a leukopack. So you keep your reagent costs down, you keep your process development costs down in the preclinical phase, but you get a very high throughput process development tool. And here's the kicker. You're doing that preclinical process development on the very same platform that you use to meet all of your clinical production needs and the commercial manufacturing needs. It's the same consumable cartridge, the same instrumentation, the same software. That is not what happens today. What happens today is typically academics develop a cell therapy process. They push it into early into the clinical phase. Then it either gets spun out into a startup or it gets out licensed to a pharma company. And the first thing they do is they look at it and like, well, this is brilliant, but it's not very manufacturable. It's very academic benchtop process. We need to make this more industrialized, more cost efficient, more scalable. So then they change out a few of the manufacturing equipment. The moment you change any of the manufacturing equipment, it triggers so much process development work and so much comparability studies that it keeps departments busy for years. So if you've got a platform that you can use all the way through from the preclinical phase into the clinic, into commercial scale manufacturing, you're eliminating years of work, which means you can get to FDA approval faster. That takes risk out of the equation for any cell therapy developer. And it's a big boon to patients if life-saving cell therapies are becoming available earlier. And it's also very valuable to pharma companies, right? Every year of on-patent production time for these cell therapies is worth billions of dollars before loss of exclusivity occurs and the drug goes off patent protection.
That's actually a great segue as we think about this. Where do you feel Solaris fits into the biomanufacturing stack? Will it be owned by the hospitals? Will it be owned by the pharma companies and sit in medical centers? Will there be a collaboration connection with the early stage facilities? So you have those instruments going all the way across the board, places like Landmark Bio in Boston, Discovery Labs in Philadelphia, that are early stage GMP facilities for exactly the development you're talking about. I'm just curious to understand because it sounds like there's so many places where the technology will be a valuable slot in.
Yeah, it's a really good question. I should say first and foremost, from a technology perspective, we're agnostic to the whole debate around centralized versus decentralized manufacturing. Are we going to be manufacturing in centralized manufacturing facilities or right at the bedside at the hospital? From our perspective, we can deploy cell shuttles in either scenario. So it really applies to both. And arguably that's not for us to figure out. We'll follow the lead of our customers and whatever they want to do, we will support. We've taken a very collaborative approach from the very genesis of this company. And I think that's the way to go in this time and day. It takes collaboration, it takes partnerships to be successful in this space. And ultimately, if our partners are successful, then we're successful. So we're going to follow their lead, I would say, when it comes to that. And we're certainly flexible in terms of what we can support. I guess one example of how we've been collaborating with the industry to date is very, very early on in the trajectory of the company. We created the Early Access Partnership Program. And that program was really a win-win situation for both Solaris as well as the pharma companies and biotech companies that we collaborated with. in that it ensured product market fit. But participants in the past included Fred Hutchinson Cancer Research Center, Pact Pharma, Poseida Therapeutics participated, and then there's one larger undisclosed pharma company that we haven't been public about. And what all of these companies did is they shared with us detailed documentation of their processes down to every critical quality attribute, every unit operation. And in this way, we got insight into a whole number of different cell therapy manufacturing workflows, autologous workflows, allogeneic workflows, CAR-Ts, TCRs, you name it. And that really put our scientists and our engineers into the position where they knew exactly what they had to build to actually create a product that has the flexibility to support a wide variety of different cell therapy modalities. So collaboration is key, and here's one example of how we did that.
And it sounds like you've really taken that customer-centric approach to make sure the technology is as readily applied as possible, just as you described. And when you talked about earlier as well, the digital twin technology for process development, I suspect that also cuts out the authoring of a book every time. So that way, instead, the consistency of the process, you can take and mix and match, as you were describing, to do almost a multiplex testing and understand which process will be most effective for that particular therapy.
That's exactly right. I didn't speak a whole lot about software. So the software is like the third pillar of our cell therapy manufacturing platform. And it really allows customers to do four things. It allows them to define their own cell therapy manufacturing workflows, do digital process development in the software platform. It then allows them to execute those workflows fully automatically on the cell shuttle. And it then further allows them to monitor all ongoing workflows in real time. And finally, you get a powerful reporting solution that can actually generate electronic batch records, mostly automatically. Maybe diving into each of these points a little bit more in depth. The Process Design Studio gives you the ability to actually design your own workflows, and that's a very differentiated value proposition in this industry, in this market landscape today, in that it allows process development scientists at pharma companies to actually do their jobs and do process development without having to go through our software development teams. They can do it themselves at any computer in their network, pull up that software platform, and 10 minutes after they're done, kick off a new workflow on the cell shuttle. I talked at length about automated execution of the workflows. What I didn't mention is we've actually automated the act of taking the sample. Sampling, the way that's being done today is you've got a human walking over to your benchtop instrument with a syringe and the Luer lock fitting, or a needle and a septum, and needless to say, that introduces the risk of contamination. So no more humans and walking over to instruments with syringes to take in-process samples. We've automated that with a technology that enables very sterile and metered liquid transfer. And then once you've taken the sample, you've got two choices. You can either analyze those samples on any third-party instrument of your choice. So we recognize every pharma company has all of their flow cytometry assays dialed in on their favorite flow cytometer, and they're free to do that. We're not locking anybody in. And then on top of that, there's also an onboard QC solution that can run 16 different assays in less than 5 minutes on a tiny sample, 0.3 milliliters. That gives you cell count, density, viability, metabolites, nutrients. All of this data then shows up in our online monitoring platform. So you get very rich in-process data. And that then further enables several additional exciting applications. To name but one, well, that's what you just pointed to, Chris. It enables the generation of electronic batch records. So by virtue of the fact that the full process is automated, our software knows what it did to the product at what time. That's all the information that goes into an electronic batch record. And then further, having readouts from the cell counter, for example, is also information that goes into electronic batch records. So that's what puts us into a position to not only transition the industry from paper-based batch records to electronic batch records, but also auto-generate a lot of that content. So the documentation burden goes down and we don't have to write a whole book for every patient dose produced anymore. A second very exciting application is that you can do closed loop expansion. Let me elaborate on what I mean by that. Typically, expansion is the longest processing step in the workflow of most cell therapies today. You're just waiting for the cells to divide and grow. And the way it's defined for most cell therapy workflows today is we're going to expand for X number of days, say 7 days. And really what you care about is not time. You don't care how long it takes. What you care about is total cell count on harvest day. You want to make enough, you want to have enough cells to make dose. And there's donor to donor variability. For a fast expander, patient whose cells are dividing more quickly, that may take a few days less. And then for a slow expander, it may take a few additional days. And what you can do with our platform now is you can define a workflow where you're expanding until you hit target cell count. And you don't define it by time anymore, you define it by target cell count. And if it takes longer, it takes longer. If it takes a few days less, great. Now you shorten vein-to-vein time for that patient and you get a less costly manufacturing process. So there's all these optimizations. And then the third point here is really we're generating so much in-process data. That data is valuable in and of itself, where you can actually start mining this data and ultimately start drawing conclusions between in-process data during the manufacturing process and clinical results. And having very large data sets and collecting a lot of data in a fully automated fashion will enable very important conclusions that ultimately hopefully will lead to better cell therapy products and also enable machine learning algorithm applications where you mine that data to draw those conclusions.
And ideally, this will also help with the requirements of scale that we were talking about earlier. If the FDA is, and we're obviously just projecting, but say producing or approving 10 therapies per year, after 10 years, you've got 100 therapies on the market. That's millions of doses that need to be produced for patients. And as a result, scaling that, especially in a decentralized way, will become more enabled as you're able to assess and rapidly optimize these processes.
That's exactly right, Chris.
All right. Well, I could keep asking questions forever, so I'm going to pass it over to Michael just for a moment so we can talk more about how you've gotten here in terms of some of the challenges and accomplishments.
Absolutely. And just to reiterate, it's truly incredible how intentional all of the designing has been. Really is a great example of product market fit done right, done up front. And as Chris had mentioned, really future-proofing Solaris and all of the solutions to add significant value kind of regardless of the direction that the cell therapy markets kind of move into. So wrapping all of a bow on all of this, would you mind sharing some of the challenges, the key challenges that you have had to have gone through to build Solaris to where it is today?
Yeah, well, I'd say one notable challenge is that our company grew up in the middle of the pandemic. And I would say more than anything that introduced two challenges. One, we had a lot of our team work remotely for long stretches of time. And thank you to Zoom for actually enabling that. I think we really mastered that challenge well. And I would actually say, while it forced us to become creative in exactly how we're working together as a distributed team with only core group in the lab actually developing the technology for stretches of time, we got a big productivity boost out of it. I remember several of our employees coming up to me and saying, Fabian, Solaris saved my life. I was so bored out of my mind. There was nothing else to do. And work was like the most interesting and exciting thing in my life. When I would usually go to a bar at 7 p.m., I would just keep working for a while. And So I think we benefited from this challenge. And I guess one other one that I can mention is the entire world has been experiencing supply chain disruptions, delays of components. Now we're building an instrument that contains a lot of components, and luckily, due to the experience of several of the leaders at Solaris, we had the foresight to actually order a lot of the long lead items at the very beginning of the pandemic. Like, all right, these are long lead items already. This is not going to get better with COVID-19 and shutdowns all over the world. So we placed those orders even earlier than we otherwise would. And in that way, we're able to minimize any kind of delays when it comes to product development.
Well, and having seen how long it's taking to, pull ourselves as a society out of the pandemic, I'm sure that that foresight has been really paying itself over tenfold. So, taking that question one step further, what have been some of the greatest accomplishments that you faced throughout this?
Yeah, I would say the greatest accomplishment, and you might think we built a product that works, but it's actually the team. If you think about what is a company, it's a group of people working together, and I'm very proud and grateful to be able to say that we attracted top-notch talent across the board, and especially last year, we put a lot of very experienced leadership in place. There's more than 24 leaders at Solaris, and all of them are incredibly experienced.
So what's coming next for Solaris?
There's a lot more in the pipeline than we've been public about. Specifically, there's a few new products, a few new services, but won't publicly disclose at this point in time.
Absolutely. And thinking more broadly, are there any additional enabling cell therapies or technologies that you're excited for?
Yeah. So what we're doing with the Cell Shuttle is really debottlenecking manufacturing. Manufacturing is typically depending on your cell therapy process, it takes between 1 and 3 weeks. And then once you're done, and you've actually got a plastic bag full of cells that contain your final drug product, now you start release testing. Release testing today is a very involved process that you end up running a whole battery of tests on the final product. And that is also a process that is not exactly scalable. So technologies that actually automate release testing are certainly an enabling technology that I'm excited about.
Maybe you've already answered this in part, but how do you think then about the transition towards long-term, hopefully, allogeneic cell therapies as we transition from autologous?
Yeah, first I should probably mention I don't really have a horse in this race. Our technology applies to both autologous and allogeneic cell therapy manufacturing approaches. So that said, I think there will be a place for both. I've had this conversation a few times with Professor Dr. Carl June, who's on our board of advisors. Carl June, for those who don't know, invented the very first CAR T-cell therapy that he then later out-licensed to Novartis. Novartis commercialized it under the brand name Kymriah, and that was the first FDA-approved CAR T-cell therapy in 2017. So had this conversation with Carl a few times, and there appears to be consensus amongst many people in the industry at this point that there will be a place for both autologous and allogeneic cell therapies because of their distinct advantages and disadvantages. Autologous cell therapies have a better safety profile. You cannot go safer than your own cells, and they also have the advantage that the therapeutic effect tends to last a long time. So Emily Whitehead, for example, one of the first patients treated with Kymriah, she recently had her 10-year anniversary, being 10 years cancer-free, and arguably that is owed to the fact that she still has genetically modified CAR T-cell therapies circulating through her bloodstream and keeping her safe, keeping the cancer from coming back. With an allogeneic product, you've got the risk of graft versus host disease that can lead to complications that in the worst of cases can lead to death. And you also have a therapeutic effect that's limited in time, because the residual patient immune system eventually gets rid of the foreign allogeneic cells. That said, they're more cost efficient to produce. And there's also a benefit in terms of how long it takes to deliver an allogeneic product as opposed to an autologous product. So, in an ideal world, just from a patient perspective, when you and I eventually get cancer, if you're not going to die from anything else, eventually all of us will get cancer. Ideally, you get diagnosed with a certain kind of cancer in the morning. In the afternoon, you get an allogeneic product that delivers immediate benefit to you. And meanwhile, we start manufacturing an autologous product that's ready a few weeks later. Well, we'll see how it plays out. There's also a trend towards shorter and shorter autologous side of things. So I don't think it'll take quite as long to manufacture autologous cell therapies in the future as it does now, nor will it be as expensive as it is today. I think the technology that we're bringing to the market actually has the potential to shift the balance towards more autologous products, because it'll be so much easier to manufacture autologous products at scale and so much more cost efficient.
We normally try to cap things off with a few rapid fire questions, with one often being asking you to imagine what, in this case, biomanufacturing would be like in 2050. But it sounds like you've just tried to answer it for us already. So I guess maybe we're asking instead, where will Solaris be then?
There are huge first mover advantages. So cell therapy manufacturing today, from my perspective, is really an unsolved problem. And whoever proves that they can solve this problem, that is to say, enable cost-efficient, reliable cell therapy manufacturing at commercial scale, whoever reaches that point first will win, I believe, most of the business. If you put yourself in the position of any leader at a pharmaceutical company, if a certain technology has been proven to work and checks off 90% of the boxes, why would I go with an unproven second mover technology? So the way I see it, we're poised to actually get to the market first. As far as I'm aware of this, there's nobody else that comes even anywhere close to where we are from a technological perspective. So if you're asking me what does the future look like in 2050, our technology will be the de facto standard for cell therapy manufacturing, and ultimately millions and millions of doses over the course of the years, and many cell therapies, millions of doses of life-saving cell therapies will be produced on our technology.
And I love that confidence in a founder. It's exactly what brings a startup and a company forward. So we're going to keep rooting for you ourselves here as well. And as we think about tech bio more broadly, this integration of technology to enable bioengineering platforms, just as you're building, that generate this wealth of information that in turn become a feedback loop and generates ever more optimized processes or early stage R&D, whatever the case might be. We're seeing increasing requirements for interdisciplinary teams. And you were speaking earlier about how perhaps Solaris' greatest accomplishment is its team. So how have you thought about building an interdisciplinary team and culture, especially given your own academic background?
Yeah, I would say that is actually one of the ingredients of and part of the secret sauce that allowed us to move so quickly, so fast. We have a wide range of different disciplines under the same roof. So on the scientific side, we've got a process development team, an analytical development team, manufacturing science and technology teams, quality teams, people with manufacturing experience. On the business development side, we've got folks who've been in this industry for more than a decade and really know a lot of the players intimately. And then on the technology side, there is some, there's mechanical engineering, electrical engineering, controls engineering, systems engineering, and at least 3 different software teams. So that makes 8 technical disciplines, and I don't know how many I mentioned across the rest of the business. And all of these teams are staffed and then led by very experienced personnel who work for each discipline. I think we managed to find true experts and exceptionally talented and experienced folks. So give a few examples on the scientific side. When it comes to cell therapy, we've got a lot of leaders from that previously used to work for Novartis. We've got people on the team who actually manufactured Kymriah. We've got people who did the analytical development for Allogene's cell therapies. Several team members who worked for cell therapy CDMOs, as well as other life science technology companies like Agilent, Finesse, Thermo Fisher. And then on the technology front, we hired some amazing talent from Berkeley Lights and Tesla, Terumo, BCT, Zymergen, Varian. Varian is making very sophisticated medical devices for proton therapy. And you get the idea. And a lot of different disciplines staffed with really experienced, top-notch people.
And I can see why you say the team is the greatest accomplishment. So to wrap up, any other closing thoughts, shameless plugs you'd like to share with the audience?
Well, I'm going to segue here and say, come join us. We have assembled a team that is exceptionally competent and also very, very passionate about curing cancer. And on top of that, a joy to work with. The entire team is extremely collaborative, and I think for a lot of people who are early in their career, that presents an opportunity to find amazing mentors. And for everyone who's very advanced and already has a long track record of how they achieved above average results compared to their peers, they'll find amazing colleagues to work with. I keep hearing that every time we hire somebody new. It's like, damn, Fabian, I've never worked with a more competent team. There's a lot of power in this team. I will also say we've been very deliberate and intentional about company culture. We always say one bad apple can ruin the whole bunch. So we're very protective of that culture to actually keep it a culture that is a joy to work in. And we also, this has been the mantra from the very beginning. If at the very end, we're successful in the sense that we deploy this technology to the market, and patients will benefit, customers will benefit, investors will benefit, but we all hate each other, that's not success. It's got to be fun. It's got to be fun along the way. And a few glimpses here. Core values, integrity, passion, and the desire to really have an impact on an industry that matters. And in our case, there's a direct impact on making cures for cancer manufacturable at large scale, which will quite literally save patients' lives. We really value people with a growth mindset and innovators who are great at collaborating and do high quality work and are just incredibly competent at what they do. So if that resonates with anybody who's listening to this recording and made it to the very, very end, please don't hesitate to reach out. We're always hiring great people.
I think that's a message that will resonate with many, I know, ourselves included. So thank you again, Fabian, for a phenomenal episode, a fantastic episode. We're very grateful for your time and your willingness to share the Solaris story and mission. Thank you again.
Awesome. Well, thank you so much for your interest. It was a pleasure being here with you today, Chris and Michael.